Hi, welcome back to Introduction to Genomics. Today's lecture is going to be about single molecule molecular biology. Now, this is not strictly speaking genomics in the sense that we're looking at the whole genome, but in terms of sort of the quantitative nature of the biology that we're looking at, it is, and so we've included it here. And hopefully, as technology continues to improve, it'll get closer towards the genome scale as well. I want to say that we're going to focus most of the lecture on specifically single molecule analysis of RNA. But there is certainly single molecule analysis of DNA. So probably the first case to think about a karyotype would be the ultimate single molecule analysis. And we'll talk a little bit at the end about some methods used to look at proteins as well. Okay, but so why, sort of fundamentally, single molecule analysis of RNA for expression is about counting RNA molecules. And so why do this? Right, so one reason is sensitivity. If you actually remember, the alternative is to get some analog readout, like the expression in this cell is 3 times as much as the cell as in some other cell or population of cells, plus or minus a little bit. So with an actual count, you get extreme sensitivity. You can potentially identify 0 versus 1 copy of that RNA in a particular cell. Accuracy, sort of for similar reasons. So you get really high accuracy. If you have a method that's very sensitive, you can actually count exactly the number of molecules in the cell. That's how many there are. It gives you really fascinating information about subcellular localization, as we'll see with some of the imaging-based methods in particular. So RNA is an especially good substrate for this because its sequence gives you a completely generic way to target particular genes. And then, so absolute quantification is sort of the overall term that we'll use to describe this. And so what is absolute quantification, and what are its benefits? So absolute quantification is just having the actual number of RNAs in a particular cell, and that allows you to really compare directly between labs. So you don't have these issues of, well, if a microarray run in this lab is different from a microarray run in that lab, and you can't directly compare them. It's just a number, right? It does not depend in principle at all on the experimental method that's used to generate it. Although, of course, it's important that the experimental method be comprehensive and sensitive enough. So it's really useful, it turns out, for computational modeling, and I'll show an example of that towards the end of the talk in the understanding of transcriptional mechanisms and transcriptional noise. And it allows really numerical inference. So what are the methods for single molecule quantification? So we're going to talk first about some of the PCR-based methods. There's single cell RT-PCR, and then which is not strictly speaking a single molecule method, and we'll extend that to digital RT-PCR, which is both a single cell method and really can be thought of as a single molecule method. And then we'll move beyond those to some of the imaging-based methods, in particular both the fixed imaging by in situ hybridization type approaches and live imaging, where you have engineered transgenes. So here's sort of a brief summary of traditional quantitative RT-PCR for gene expression measurement. You have a whole bunch of cells, and you run them through a meat grinder, so you've lost all the spatial information that was in those cells. Or maybe you have something slightly more sophisticated than a meat grinder, but serves the same purpose, and then you do some PCR. And you have basically the PCR reaction versus time measured in a number of cycles, and the earlier that PCR product reaches a particular threshold, the more starting material template there was in your population of cells. And so you can get quantification this way, well within twofold in terms of the reliability of the measurement between individual samples, which is, of course, you can have much higher levels of biological variability. But how can we look at single cells? And so first, of course, you have to have a way to get single cells, and these are sort of the major approaches that are used for that. Obviously, you can just take a bunch of cells if you have them isolated so they're not stuck to each other, and just dilute them out until you have only one cell or less in each tube. You can also go in with a needle or some other kind of apparatus and actually manually pick up the individual cells, again, in particular if they're especially well separated. I have a picture here of sort of an example of something called laser capture microdissection, where you have a collection of cells where you basically use a special resin, you hit it with a laser, and then that allows you to selectively pick up just the cells that you hit with the laser, and so you can identify either individual cells this way and purify them, or you can get particular groups of cells if that's what you're interested in. And then the last method is flow cytometry. So again, the way a flow cytometer fax machine kind of works is you have a bunch of cells going through a tube. Use a laser to figure out whether they're fluorescent or what their scattering characteristics are. And then you can have particular gates. So if it fits a particular set of criteria, in this case we're getting the red cells, you can put the red cells into tubes. And if you want to, you can put one red cell in each tube. And so it's another way to get single cells. Now, once you do this, you have to actually do an RNA prep and convert the RNA into cDNA, and then you can do your PCR reaction, your quantitative PCR reactions. And so it's inevitable you're going to have some loss of molecules during that purification process. Nothing is 100% efficient. So that's important to keep in mind when you try and interpret results from this kind of experiment. So you take that cell, you lyse it, you make cDNA, and you use the qPCR to amplify it, and you get data that looks like this. Now again, just as a reminder, they have the PCR going along this way, so this is time, and at some point you hit a threshold cycle where you start seeing an increase in fluorescence, and the number of cycles it takes to get to that threshold cycle is related then by a simple exponential process in theory to the number of starting molecules that were in your sample. And so you can back calculate and figure out how many molecules do you think were present in the original sample and compare across samples. And this has, you can actually take this down to the level of single cells. No problem, especially for reasonably highly expressed genes. Once you get to lower levels of expression, it becomes more problematic. And it has some really important advantages, and one of which is that you can do multiplexing. You can have different qPCR primers in the same reaction, and maybe label, for example, with different colors. There's particular types of probes that have different colors for each kind of target. You can also just do lots of different experiments. They have qPCR machines with 384 well plates, and you can just blast through tons of cells, tons of different genes in those cells. So it's pretty cheap to do any given individual assay. Certainly by comparison, you get a really high dynamic range. That threshold cycle is in a log scale. So when you compare between threshold cycle of 20 versus 30, you're talking about 1,000-fold different right there, and that's you have even more than that. And you can compare, for example, if you have a single nucleotide polymorphism in the cells that you're generating your data from, you can look at allele-specific expression. You can look at different splice forms, and just by designing appropriate primers and probes. So pretty powerful technique. However, it doesn't really get you down to this individual molecule counting approach. Right, so people have done some fairly careful analysis, and this is just one of them that's cited here, where the bottom line of this is just sort of measurements of the measured amounts of RNA per cell for different genes, where they have an idea what the actual expression level is, and the bottom line is, and these arrows here are the actual error rates. So by the time you get down to genes with really low expression level, the bottom, really that limit is between, say, less than 100 mRNAs per cell. The noise starts to increase to being similar to the level of signal, and below about 10 RNAs per cell, you're sort of out of luck, pretty much. Right, so digital RT-PCR is sort of a method to try to get around this. And basically here, so instead of having one tube where you have all your RNA mixed together, you take your cell, you'd actually dilute its contents out amongst many different wells. In this case, you probably have hundreds of different wells, and you do your qPCR in each well, or your RT-PCR in each well, and you just look, was there an RNA in there or not? And so, here's a case where you have a little green spot. Here's another one here, right? So those are wells that had an RNA molecule, and if you do this dilute enough, you pretty much know that each well had 0 or 1, and you can use a fairly simple mathematics to figure out then what the total number of RNAs were in the starting population. Okay, so this is actually giving you now really a single molecule level count of the RNAs that are in that cell. And it has its own issues, one of which is the dynamic range, and that's a big issue. So if you have now the case where you have, say, 1,000, 10,000 molecules per cell, and you put those into, say, 500 wells, you're going to have trouble because every well is going to have 20 or more molecules in it. So you really have to be, you're limited in the range of dilutions that you can analyze. And these are things that are pretty complicated. You have these little microfluidic chips that have the actual wells in them. It's harder to multiplex, and it's just not very efficient to do really large sample sizes. So now moving on to imaging-based methods. So the advantage of imaging, we'll talk a little bit about the advantages of imaging-based methods. They certainly are extremely powerful. One of the leading methods was developed by Arjun Raj, who's now here at Penn when he was in Sanjay Tyaghi's lab at NYU. And so we'll talk about some of the advantages of those. Right, so the two main categories of imaging-based methods are, for single molecule analysis of mRNA, are fixed and live cell methods. So for fixed methods, you're typically using some kind of a fluorescence in situ hybridization where you have probes that are antisense to the messenger RNA that you're looking at. You can design probes with whatever sequence you want to, so they can target any endogenous RNA, and you fix the cells and you put them in there, but you don't have any dynamics there. So if you want to see how RNAs are moving across time, you can't use those methods. So by contrast, live cell methods, you actually have to go in with, so far at least, with engineered RNA. So you have to put an RNA molecule into the cell in some way that has binding sites for some kind of a probe, and we'll talk about a couple of different designs for that. But then it lets you look at the movement of actual individual molecules over time. Okay, so for the fixed methods, we'll talk about the direct detection methods, which is really fairly simple. I think you'll agree once you see the logistics of how that works. It requires a fairly high-end microscope, and it has some limitations. You can't look at really small sequences, microRNAs. You can't look at really small splice forms, allele-specific expression as well. And then some of the amplified or indirect detection approaches that have their own advantages for these really smaller cases where you really need extremely high sensitivity. Right, so this classic method for direct detection-based FISH is to use these tiled sets of oligonucleotides, and so there's sort of two approaches, and they both have actually work fairly well. And I mentioned Arjun Raj, who's at Penn, who was one of the developers of this method, where you have essentially 30 or more. It actually works apparently down to about 20 probes that are each complementary to a different part of your messenger RNA. So this is your messenger RNA here. And so you have these complementary probes, and each one has a little red dye on it. And once you have about 20 of these probes lined up, you get a little diffraction limited spot on your in your cell that corresponds to that messenger RNA molecule. And sort of the alternative method, which Rob Singer's lab developed many years earlier, was to use a smaller number of probes. So this is one of the advantages of this method, is that you actually don't need as much overall probe length. You can have a smaller sequence that you can target, for example. So you have multiple labels on each probe, and they're slightly longer, say 50 base probes instead of 20 base probes. It turns out that having those probes or those fluorescent molecules more closely spaced actually isn't necessarily that much of an advantage. They tend to sort of start self-quenching when they get really close together. But these are sort of the two major methods, and also these are much more expensive to generate than the end-labeled, singly labeled probes. So that's been easier to scale up. Okay, so you make up these probes. What you can do, there's fairly well-established protocols that you can find online to do this. You hybridize them into your cells that you're interested in, and you get an image that looks like this. So this is just an example, courtesy of Dr. Raj, where you have, in this case, you have a cell. Let's see if I can find a color that works. So this thing here, this is the nucleus, and you have all of these dots outside the nucleus. And those are individual mRNA molecules, and they can show that in part by doing, for example, approaches where they label the two ends of the RNA with with different colors and looking at their separation and relative brightness. They can do bleaching experiments, and so they think they have a pretty good idea that in general these are individual mRNA molecules. You can see there's also some mRNA molecules inside the nucleus, and there's these two bright spots. And those bright spots correspond to, and this has been again studied by a number of different methods, to most likely to the site of active transcription. Turns out if you do the same experiment but use probes that target the intron of the gene, you really just get those two spots in the nucleus for most genes. And so that sort of provides some evidence to confirm that splicing is probably happening co-transcriptionally in most cases. Okay, so you have to, given one of these images, there's some analysis that has to be done. In particular, you have to be able to count the dots. Otherwise, you don't have this absolute quantification that we've been so excited about. Right, so you have again, just a reminder, you have your gene with its probes that are stuck to it. Here's an example. And basically what it comes down to is thresholding. So you have, here's your raw image, and you just try a bunch of different thresholds. So you say if it's brighter than 20 intensity units, let's call it part of a spot. If it's brighter than 30, let's call it part of a spot. And as you change that intensity, you go to a higher intensity, you identify less spots. And what's been observed is basically there's this region here where you have, it doesn't really matter how you change the intensity threshold, you find a similar number of spots. And that's really the region where you're getting a fairly accurate count. And so by doing this, you eventually get this, you convert this image into a number in this sort of in silico spatial representation of where all the mRNA molecules are. All right, so this is just an example from worms, and this is a publication related to this. So this is a separate image in Raj et al. In Nature 2010, where they studied the regulation of intestinal specification in worms using single molecule in situ. And so each of these red dots now is a messenger RNA for the master regulation of intestinal differentiation, which is ELT2, and then the blue spots are just DAPI, so that's showing you where the DNA is. And see if I can get the movie to play. So important to realize that you have some three-dimensional information here, and you can actually go through and identify not just how many spots there are, but they're not randomly distributed. It turns out that those particular spots are located in regions of the embryo that give rise to the intestine, so that's sort of satisfying for an intestine-specific gene. So you can actually go through and try to assign which mRNAs go with which cells as well. And this is now, again, from the same type of experiment. Again, these are C. elegans embryos. You can see we have some really early embryos, and they're expressing a blue gene, so that is an early gene expressed in intestinal specification. We can multiplex. So we have the blue probe for that early gene. Here's another embryo where you have a blue gene and a yellow gene, which again is another intestinal specification gene. And then later on you see the red, the L2 coming on to sort of turn on the terminal intestinal specification cascade. So you can get 4 or 5 different genes in one sample by multiplexing this way, just using different colored probes. You also get subcellular localization out of these experiments, and so I mentioned the spots, for example, in the nuclei. So here they are again. So these are the the sites of active transcription. You can see for this particular gene, you're getting actually all of the mRNAs, or all of the RNAs that are produced are pretty much staying in the nucleus, or if they're leaving the nucleus, they're being actively degraded. So you end up with this distinct pattern where really, you have the sites of transcription and then these nuclear spots. And so this has been done, for example, to look at where non-coding RNAs may be acting. So yeah, there's, if you'll remember or you'll see in the non-coding RNA lecture, that there's all these long energetic non-coding RNAs that are being produced from many different genomes. One way to figure out what they might be doing is, well, are they in the nucleus or are they in the cytoplasm? Because if they're being translated into proteins, they're probably in the cytoplasm. If they're regulating transcription, they're probably going to be in the nucleus. And so we see in many cases anyway, they actually are in the nucleus. Okay, so this kind of approach, where I told you one of the advantages of absolute quantification is that you can actually use the precise counts for specific to ask specific kinds of questions about transcription. So this is sort of an example about from an earlier study, Raj and Tyagi in PLOS Biology, looking at how noisy transcription is, in particular whether transcription occurs in bursts. And so the model is that you could have sort of the simplest model would be you have your DNA, and at some sort of through some random process, you transcribe that DNA and you get some RNA molecules, and then some other process causes them to be degraded and lost. And as an alternative, though, you could have bursting. And bursting means basically that this DNA goes through periods where it produces a lot of RNA molecules, and then it becomes silent and goes into an off state. And it could stay in the off state for a while, or it can come out of it fairly quickly, and then eventually go back on and start producing more RNA molecules. And it turns out that if you have bursting, you actually get an extended variability in terms of the transcript count. So if you have no bursting, you have sort of a Poisson distribution, which you can figure out from the mean transcript levels. But if you have bursting, you end up with a much higher variability, and with some cells having huge amounts of RNA and some having very little. And that's exactly what you see. So this is just, now in this case, you have this cell and this cell, which both have just blazing nuclear expression of this particular transcript. This one has really high cytoplasmic as well. This cell is basically quiet, and this cell has very low levels. And so you can actually use these counts to infer not only is that there's bursting, but what the rate parameters of the bursting and the transcription are. So this is great if you have a nice long messenger RNA, say 500 bases long, 1 kb long, longer. You can generate your 30, 50, 100 probes that tile along the length of that molecule. But what if you're interested in, say, a microRNA? A microRNA is 21 nucleotides long. What if you are interested in a splice isoform where two different splice isoforms only differ by 1 exon that's, say, 80 bases long? You need to have something that's more sensitive because you can't tile 20 more probes along an 80 base exon. And so there's a variety of schemes that are used that basically you take a single hybridized oligonucleotide and then amplify the signal that comes off of that. And a lot of times these use something called LNAs, or linked nucleic acids, and those basically are these special varieties of nucleic acids that have an extra link in the backbone of the nucleic acid that sort of locks it into a state that's favorable for base pairing and base stacking interactions. It's not totally clear how the sensitivity and specificity of this method works out. It's sort of hard to directly test that. But certainly you can get results that make sense. So one of the amplification methods, and this is just sort of an outline here of the way these LNA probes work. You can imagine you have this sort of locked nucleic acid probe, where it's base pairing with this cognate sequence in the way that a complementary sequence in the way a normal fish probe would, but it has this extra backbone link that causes that interaction to become more stable. And you can use this not just, so this is a traditional mRNA where you have now stuck to the end of your probe, you have say an enzyme that you can then use to cause a inert substrate to become fluorescent. And then you get these little foci of fluorescence forming around each of the individual spots. And so this is mRNAs, and then on the right here we have the same thing done with microRNAs. And you can definitely get countable dots that vary from microRNA to microRNA by using this method. Now, how exactly these correspond, we don't know perfectly the sensitivity and specificity, but it's certainly a promising approach for this kind of question. There's also, in addition to the enzymatic amplification schemes, you can also use branch DNA or padlock probes. So branch DNA, basically, you have your first probe which maybe is still an LNA. It has these extended region sticking off the end of it, and that region is capable of binding to additional probes. So you add sort of secondary probes, roughly equivalent to a secondary antibody, and then you can go in with a third step and have additional probes binding to your secondary probes. So by binding even just your initial probe, then you're able to recruit using that probe enough fluorescent molecules for you to be able to detect it. This padlock probe is sort of another alternative. Basically, this is the padlock probe. This is inside out. Basically, this thing has these regions on the ends that are complementary to to the region that you're trying to hit. And again, this can be, you know, on the scale of, say, a microRNA or a small exon. And there's a gap there normally, but if this thing hybridizes, you can ligate those things together, and then you can use a rolling circle kind of amplification to then expand this so it gets you enough sequence to probe by traditional fish kind of methods, and you get this sort of long thing. This is what a rolling circle amplification looks like. You get this long molecule coming off of that circle that you can then probe with additional probes. Okay, so for live imaging, now remember the pros of live imaging, you can look at the movement of individual molecules over time. Cons are that you have to engineer your cells to have particular both reporters and probes within the cells. And so there's particular issues for the two main methods that are used for this, which are MS2-based methods and molecular beacons. And we'll go through each of those in turn. Okay, so here's the basic schematic of the MS2 method is that you have your mRNA, and now what you do is you take, in a transgene, a whole bunch of copies of this sequence called the MS2 binding site. So you have maybe, I've shown, I think 12 or something like that here, but in reality you might have 24, 48 of these MS2 binding sites, and MS2 that's just a binding site for a phage coat protein. So in the same cell, you express that coat protein fused to GFP, and those will just line up along this 3' UTR, and you get now this nice concentration of GFPs that are recruited to your messenger RNA that you can then detect as a diffraction-limited spot. We have a movie of that here from this molecular cell paper. And they did this in yeast originally, looking at so basically one of the neat things about yeast is they segregate this particular protein to one of the daughter cells, and allows them to make sure that they have appropriate segregation of mating type, sort of the yeast equivalent of males and females in the population. And we can get that movie. So the spot that you see there is going to be the messenger RNA. Yeah, so this spot is moving around, and then you can see it sort of eventually decide it's going to go through and go down to the daughter cell and hang out over there. Right, so that's kind of experiment that would be really hard to do without live cell imaging. So an alternative approach is something called molecular beacons, and the way molecular beacons work is essentially with a quenching kind of approach. And for sort of technical reasons, you basically have to make sure these beacons are pretty highly tuned. And so again, you have a target sequence, and the target so far hasn't been a random sequence, but instead is still a bunch of copies of now the sequence that's complementary to your beacon. And so the beacon itself is actually this little hairpin probe that has self-complementary, so it makes these little hairpins. But when you introduce that into the cell, typically you have to do something like inject this into cells that are expressing your particular your target labeled mRNA. Then it'll, instead of self-hybridizing, it'll hybridize onto your little target sequences, and you'll be able to line up a bunch of fluorophores. One end of your beacon has a fluorophore, the other end has a quencher, and so when it's in the closed conformation, it's not fluorescent, but when it opens up and binds to its target, then it becomes fluorescent. And again, you can use this to visualize the movement of mRNAs in single cells. And so here's just a picture of, you can actually watch individual mRNAs, in this case moving around within a nucleus, which is the sort of lighter gray area, and also the cytoplasm, which is the darker area outside of there. And so this is actually a really powerful way to look at just sort of the basic physical constraints on movements of RNAs within different parts of the cell. And so one of the cool things that Vargas study that movie came from did was co-label with a marker for chromatin. So that's the red marker on here. And so if you follow the green dots, which are individual RNAs, and sort of look at their position over time, you can see they sort of perform a fairly random walk. It turns out every now and then they get stuck, and they need some kind of ATP-dependent process to get unstuck. And you can actually sort of integrate over time and see where are all the places where the messenger RNAs have been relative to the chromatin over time. And you can see that there's actually sort of this exclusionary principle where regions where there's lots of chromatin are essentially not free for the mRNAs to diffuse into. can also use, again, just like we showed with the in situ kind of methods, these beacons to look at bursts of transcription. You can actually actively watch a particular burst happening in real time, and this is just sort of a schematic over time where you have the first transcription focus developing, and then you can see gradually over more time, more and more mRNAs sort of accumulating until you sort of hit the peak of the pulse sort of down in this region here. So you can actually actively observe these things happening, not have to infer them from Poisson. Right. Okay, so the downsides of these approaches is obviously you have to engineer these mRNAs into the cell. You can't just use probes that target endogenous mRNA. There's some artifacts now. You imagine that you stick 48 copies of a particular sequence on the end of any mRNA, you might interfere with its normal localization, and that definitely happens in some cases. And so you have to think about maybe using some kind of a fixed cell endogenous probe-based method as a corollary to make sure that your results are right. And there's also issues with the delivery. So especially in the case of beacons, where you actually have to do something like inject them into the cells, it's really not a very efficient process. Right, but there's lots of examples of important biology that have come out of the single molecule live imaging. One of the issues is burst, and I showed you some examples of that, and those were really pioneered in the studies I cite here. There was a study that looked actually at the way that mRNAs are exported from the nucleus, so you can actually see mRNAs go up to the nucleus, play around for a while, and actually it takes them several tries before they actually make it through the nuclear pore. And then there's just the fact that you can explain a lot of mRNA's movement by essentially random Brownian motion. So for the last part of this lecture, we're going to talk a little bit about protein molecules. So visualizing single proteins is a lot harder than single mRNAs for the same reasons that doing anything with proteins is harder than with mRNAs. They just don't have the generic advantage of Watson-Crick base pairing to allow you to probe them in the same way. So you can't tile a bunch of probes along the protein molecule and just line them up. If you have a single, say a YFP fusion to a particular protein, the thing is the proteins are moving around so fast you can't just take a time-lapse picture unless you have some way of keeping that protein from moving around, because by the time you get enough photons for you to detect it, it will have moved somewhere else in most cases. And so, in general, you sort of need really sensitive microscopes, and so TURF is sort of the classic one that's been used for this kind of purpose. And alternatively, methods to tether your proteins to keep them from moving around too much or keep them in defined locations and to allow you to average over time. Now despite those technical issues, this certainly has been done. So one of the sort of take-home messages is they use the brightest possible fluorescent proteins. So these early studies used variants of yellow fluorescent protein, Venus being sort of the most famous. There's newer ones now, which have the highest amount of brightness of any fluorescent proteins. You have to use, again, these TURF kind of microscopes and find ways to integrate over time. And so if you look, for example, now we're looking at a lac promoter output again, a little bit different experimental setup. And these are individual E. coli molecules. This is actually under a condition when the lac promoter should be inactive. One of the interesting questions about the lactose promoter is that it needs lactose to be active, but you need the lactose permease gene is actually part of the lac operon, so you don't express any of the molecule that lets lactose into the cell unless the lac promoter is active. So it's sort of this paradox where how do you ever get enough lactose in to turn on the expression of the permease? And the answer is there's sort of this stochastic sort of bursty expression of the permease itself. And this is one of the ways that this was established. You can see now these molecules are off, but as you look over time, they'll begin, one of the bacteria, and several of the bacteria will sort of burst on little bits of the yellow fluorescent protein. And so you can see sort of little bits coming on and off. And this is not a movie, I don't think. So it allows you sort of to determine then if you add a little bit of lactose, basically the cells are off because they don't have the permease, and then as soon as one of them turns the permease on, it'll go full throttle, and that'll allow it to kick the entire pathway into high gear. Okay, so another method that's been used a lot, or at least more than many, for single protein level analysis is called optical trapping, most notably by the Block Lab at Stanford. And so this is one example of a study of that type where they actually tethered. So basically, the way optical trapping works is you're able to get these beads, which are denoted in blue here, and by appropriately focusing a little cone of light, you can actually keep them immobilized. And if you try and pull those little blue beads out of the cone of light, it actually exerts a attractive force to try and pull it back in. And so you can actually then tether, say in this case, RNA polymerase onto one of those beads, a DNA onto the other bead, and you can allow the polymerase to sort of transcribe along the length of the DNA. And if you do this, in this case it was sort of cool, they did it with different limiting nucleotides, and so they could look at how long the step size was between. They could actually measure individual steps of the polymerase along the DNA, and when it took a long time, you knew that it was at a nucleotide that corresponded to your limiting nucleotide. And by doing this with all 4 nucleotides, they could actually get a sequence of this molecule. So this has been used for a variety of sort of mostly biophysical studies to look at how things like motors or polymerases actually work in this processive way. And the last method I want to talk about for single protein analysis is something called DNA curtains. And the way these things work is basically you have along one end some little attachment areas. And so those are denoted here with this A. And so you have your DNA stuck to these anchors. And then if you either use flow or electric current, you can actually then cause the DNA molecules to stretch out towards, for example, your area of positive charge, and you get these stretched out arrays of DNA. So that's something called a DNA curtain, and this is what they actually look at if you stain them for a DNA dye. So the green is just the DNA now. Each of those little lines is one DNA molecule hanging down, and you can go in now, if you're doing this in vitro with particular, say, quantum dot labeled individual proteins. So now if you make your protein in vitro, you can put a really bright fluorescent molecule like a quantum dot onto it, and you can watch as these proteins move up and down. For example, this FITS-K is a DNA translocase, so you can actually watch the nature of how it moves. This has been used also, for example, to look at particular DNA binding proteins sliding up and down on the DNA, and if you actually put, for example, a nucleosome there, it'll stop and can't go through the nucleosome, as one example of how this has been used. Okay, and I didn't want to dwell on it. We've talked before about next generation sequencing, but actually a number of the next generation sequencing companies are actually using single molecule methods. The pioneer here was Helicose, which was actually using methods fairly similar to the big players, Illumina solid type sequencers, but doing the imaging at the level of single fluorescent molecules. Current players, Specific Biosciences, uses sort of a just a little bit different approach, but they're actually measuring individual, the incorporation of individual fluorescently labeled nucleotides into the mobilized polymerase. So they stick the polymerase at the bottom of a well and then watch as the different nucleotides come in. And then Oxford Nanopore and really a whole suite of other nanopore-based sequencing companies are using sort of actually passing individual DNA molecules through these tiny pores and measuring basically the electrical signal across the pore as a way to get the DNA sequence out. So those are all single molecule methods as well. So just as a summary, now I think single molecule approaches are becoming more and more powerful. You can look at both in cells, live cells, fixed cells, and in vitro, and really look at the distribution of especially mRNA molecules. So this is really allowing absolute quantification, which really lets you ask questions in a much more quantitative way than you can with more bulk methods. You can also learn a lot from live imaging just in terms of how mRNAs are generated, metabolized, localized in real cells. And while single protein imaging is challenging, it is definitely coming along. Anyway, thanks, and we'll see you next time.